Thank you for joining this webinar. Today we're going to be discussing specifically the viability assay on the Celigo instrument that utilizes Hoechst PI. In this assay, Hoechst will be used to stain and determine the total cell number, while the propenium iodide will be used to determine the number of dead cells. So we have a full seminar for you that will really touch on many different areas. We essentially are going to start today with an overview, and in this overview you will see just some of the things that we are going to discuss. We'll start with assay setup that will briefly talk about plating, the number of cells, and staining the cells. We'll then talk about the Celigo setup, how to actually acquire the image, perform image analysis, and then view the results data. We'll also go into a little bit of the Excel for percent gating and total cell counts. So for the initial lab setup, you'd like to first get a cell preparation going. So you want to ideally have a 95% confluent flask, remove the media, and wash it with PBS. You then want to trypsinize your cells, presuming that these are adherent cells at 37 degrees in the incubator. After you've finished trypsinizing, you want to spin the cells down and obtain a cell count. Once you obtain a cell count, we really kind of want to make a cell map. And this particular cell map that you're looking on the left goes from 40,000 cells per well all the way down to 20 cells per well. To do the serial dilution, this is just one of the suggested ways that you can do it. You can create a suspension of cells that are approximately 400,000 cells per mil, or 400 cells per microliter in a 2 mil solution. And then you take 11 more tubes with 1 mil medium, and you pipette 1 mil from the tube 1 into tube 2, making a 2x dilution. Therefore, you will approximately have 200 cells per microliter concentration in the tube. You serially dilute in this manner all the other tubes, and then you pipette 100 microliters of the media with cells into each 96 well plate. We recommend that you gently spin the plate in order to settle the cells in the bottom, and then you incubate the plate overnight to allow the cells to adhere. For the staining protocol, you will need Hoechst 33342, which is cell membrane permeable, and propenium iodide. Both of the reagents are provided by Nexcelom Bioscience. Additionally, you need cell culture media, conicals, pipettes, and multi-channel pipette, as well as a reservoir. We are first going to make a 2x staining solution. We make this by taking a 15 ml conical and adding 12 milliliters of cell culture media, 10 microliters of Hoechst, and 24 microliters of propenium iodide. This is now a 2x master mix. You use a multi-channel pipette to dispense 100 microliters into each well, making a 1x stain solution. So if you remember previously, there was 100 microliters from the previous evening with your cells and media, and we're adding 100 microliters of this master mix solution. We would like to then to incubate these cells for 30 minutes at 37 degrees, and then go ahead and image in the Celigo. Next, we'll talk about the Celigo setup itself. First thing we need to do is we're going to create a new scan. So for those of you that haven't really done this before, this is a very good introduction of how to do this. You first select the plate type, 96 well plate, and then you select the manufacturer and the product number that matches your corresponding cell. In this case, it's 655090 Griner plate. You need to put in a unique plate ID that is going to be used as an identifier for that particular plate, and you click Load Plate. You'll have a pop-up window that says once plate has been loaded, click OK. By clicking OK, this will load the plate. We have now moved from the initial part to the scan section of the software. This is where the setup occurs for setting up your imaging parameters. The very first thing that we must do is select the assay. In this particular case, we scroll down to see the dead plus total. So the dead, as you had might guess, is going to the PI positive cells, and the total is going to be the Hoechst positive cells. So we first have to select an assay, and in this particular case, under the cell viability tab, we select the dead plus total. In the dead channel, it's going to correspond to the red, and now we want to switch it to the blue channel, which is the total channel. Okay, and you can see that it's a blue excitation. It has a default illumination of 10,000 microseconds, but for now that won't matter. You click on snap, and by clicking on snap, you first see the cells. Although they're very dim, it may be very hard to see on your screen because they're hard to see on my screen. We will increase the exposure time after we finish focusing, but in this particular case, you just click snap so that you're not photo bleaching your sample. Other alternative is clicking live and having the LED on at the same time, but there's a higher risk of bleaching your sample. You can then, as you can see on the right-hand side, where my cursor is, you're currently in well 1A. You can use the wheel on your mouse to zoom in or scroll around the well. And then we typically like to go to well C3. This is somewhat a little bit in the middle of the plate that allows a more uniform image to have. So we're going to click next focus setup. This is, you have to do this in order to set up your focusing for all subsequent wells. We're going to use hardware autofocus, and we're going to press register auto. By registering auto, the machine automatically selects the best cells in focus and sets that focal plane. To close it, you click focus setup. We're now going to increase the exposure time in order to actually get some of these blue nucleated cells in better, sharper, more bright focus. We need to get this intensity now down where you can see my cursor is somewhere between 100 and 150. So we're going to continue to increase the exposure time from the initial 10,000 microseconds to 50,000, 40,000 microseconds until we're satisfied that the intensity that is showing on the screen is going to be somewhere between 100 and 200, 100 and 150. Don't worry about the super bright ones that have a very high extension number. We're looking at a general population that should have that intensity range between 100 and 150. So in this particular exposure time is okay. So we've completed the focus setup, the illumination, and the exposure. We're now going to move over to the dead channel, which is the red channel, propylium iodide channel. Once again, we're going to click snap to take a look at the image, and we really don't see anything. Okay, so it's difficult to say because a lot of these cells are live. There's very low population of dead cells, so I'm going to move around until I find some dead cells here. I found a few dead cells, and using those dead cells, I can find them and put them in focus. So the instrument, once again, will take multiple Z-stacks and pick the best in-focus plane cells. This corresponds to currently 12 microns as an offset. So by clicking the offset buttons, the instrument, upon acquiring a blue channel, will then move a 12 microns offset and then acquire the red channel, therefore assuring that the red channel and the blue channel are always going to be in focus. Okay? We once again want to make sure the intensity is good, and then once the intensity is set up, we are essentially ready to image our plate. Okay? So we have multiple dead cells here. We have set up both the dead channel and the total channel, and now we can go on the right-hand side and click selection, and by clicking the box between A0 and A1, you will select all wells, and you can press Start Scan. We'll now move from the Scans tab to the Analyze tab. As you can see, some of the green wells, some of the wells are starting to appear green. Those cells that are green have already been scanned and are ready to be analyzed. You can go ahead and click on a well, in this particular case A2, and we have some initial parameters that you can see on the left-hand side that are automatically set up. And here on where you see me scroll and click is I'm turning on the graphic overlay. This shows the outline. We're going to select the well mask, meaning that 100% of the well mask is going to be counted. We're going to switch the channel to total because this is looking at blue cells. And these are some two parameters that are most likely we're going to be playing around with: the intensity threshold and the cell diameter. You can also click Check Auto Analyze in order to automatically change and update those parameters instead of having to click the Analyze button each time. Here you see some cells that were not counted very well. By pressing separate touching objects, the system automatically segments them into multiple cells. You can then click as well and look at the dead cells, total cells, as well as dead counted cells. So in this particular case, I'm showing you that blue color outlines PI positive dead cells. We're going to scroll around to make sure that all cells are counted, and it seems like some of them are not counted and some of them are very dim, so we're going to decrease the intensity threshold. By decreasing the intensity threshold, we're able to pick up more cells. Okay? By decreasing the intensity threshold, we are now picking up some of these smaller cells and smaller debris as well, which we will deal in a little while. So we're going to play around with the intensity threshold until we get the diameter intensity threshold that we like. Additionally, cell diameter is an average value that the object expects it to be. So in this particular case, we see that it's only one cell, one PI positive large cell, but it's segmented into two individual cells. So by looking at the position on the bottom left-hand corner, we know that this cell is approximately 15 to 20 microns in diameter. And by increasing the pixel diameter, we're then able to have that cell not segmented into two. We're going to continue to optimize the intensity threshold, and now we're looking at a pretty good count. There's some small cells that could be particularly debris. You can see that by clicking on the graphic overlay. So we will increase the cell area to perhaps remove some of these smaller, non-specific or debris staining. We're going to increase the cell area to approximately 25, and as well as to maybe even a larger area, and we're going to see if there's any overlap between the total, which is in yellow, and dead cells, which are in blue. All cells that are both blue and dead, as shown here, are going to be counted as PI positive dead cells. Let me repeat that. Any cells that are touching the blue portion, the outline, and the yellow portion will be counted as dead. Any cells that are not touching or associated with the nucleus are going to be counted as debris and will not affect your counts. We can now click Select and start Analyze. By analyzing, we are now not only scanning, as you can see on the bottom hand, but we're also analyzing the sample at the same time. These are right now representing percent dead. So you can see that you have very small percent of dead cells, from just under 1% to about 3% of dead cells. The system acquires images and analyzes simultaneously. We can now look at different settings, like, for example, a heat map, and as well as dead counts. So dead counts is the number of dead cells, or total counts, the number of total cells, which is Hoechst positive cells. By changing the heat map, we can select any value that we want, and we can say that the green cells, for example, are our low number of cells, yellow are the medium, and anything above 40,000 will be red. So as you can see, it's very easy to navigate and look at the total cell population during your dilution series, from 40,000 all the way down to few cells per well. And you have a visual representation using a heat map. You can click on a well to zoom in and look at a high definition image. And by zooming in on the wheel, you can look at the total number of cells counted, as well as those that have been identified by the software as dead. You can toggle back and forth between TED, total, or looking at just dead cells. You can turn on and off the total cells display by clicking the image display button and adding or moving off the Hoechst positive cells. This allows you to really get a nice representation of the images at hand. We can export those images into Excel by clicking Export Well Level Data. This is exported as a CSV file. So now that we have gone through the Celigo setup, let's just take a little bit of time and look at the graphing data. I've set up here a template that looks at our plate map of our series dilutions from 40,000 all the way down to 20,000 cells. I have a table for percent dead and percent total. Now let's take a look at the actual data that is exported as a CSV file. On top here, rows 1 through 13 are actually data that is saved automatically for your exporting information. here I'm also showing all of the information that you may or may not be interested in, including the fluorescent intensities and standard mean deviations. On the bottom of the page, you see analysis settings. This is the position as well as your parameters, so you don't have to worry about saving your parameters. As I scroll back up, please notice that we have a percent dead population that is shown here. We have a percent dead count, and we have a percent total count. These are absolute numbers that have been identified by the software as the number of dead cells in each particular well, or the total number of cells in each particular well. We can now copy this entire page, and we are going to transfer this data onto our template page. So we're going to do a blank page, and we're going to simply paste all. The template automatically draws data from the template page and fills in the averages and the standard deviation counts for your total and your dead cell count. It also looks at your percent dead cells for each particular point, for each particular dilution series point, and then it also looks at the total number. So as you can see, our total cell count is 40,000, but we have about 48,000 cells because these cells have grown over time. These are expected values versus actual values that have been acquired. So you can graph that, and you can get an R value of expected versus total, which is very close to 1. And the dead cell population is between 1% and 3%, and you can see a nice little trend throughout the entire plate. So looking at the expected count, we can map and graph all of the cells linearly. So using this template is useful for the future of setting up the experiments. So in summary, we looked at the assay setup and the plating. We also looked at how to acquire an image. We also looked at the results section as well as data analysis. I know that this has been a whirlwind of a seminar, fairly rapid fire. But if you have any questions at this time, I would love to take your question and answer sessions. Thank you.
Thanks, Dmitry. That was a great presentation. So you can see there on the screen how to capture bright field images in addition to the Hoechst PI viability. Is that confirmed for next month?
Yes, I believe so.
Okay, great. So at this point, we do have time for any questions that you might have had over the course of the presentation. Questions can be about any of the content that Dmitry just covered, but we're also open, and this is a great opportunity, if you're having any questions using your Celigo in any other capacity, we'd be happy to answer those for you as well at this time. So we'll just give everybody a moment just to get some questions going. All right, so we have our first question. Dmitry, your first question is, you had mentioned that while the system is continuing to acquire the images over your whole plate, however many wells you have, once you start receiving those green ones, you can then move over into your analysis to set your analysis settings.
Correct.
So the question here is, will it slow down your system, your Celigo, the processing, if you are trying to do both at the same time?
Yes, that's a great question. So there are multiple parameters to really think about. The speed at which the Celigo acquires those images will not be slowed down. The actual hardware in the Celigo has been optimized to such effect that really it takes images so quickly that the speed at which it takes images is not slowed down by concurrently running an analysis mode. However, if you are acquiring images and analyzing at the same time, your analysis speed will be slightly slower compared to doing analysis only with no image acquisition. So this is kind of like catch-22, because on one hand, if you want to wait until all your image acquisition is finished and then start analyzing your data, the amount of total time that you'll spend in front of the Celigo is actually going to be longer than if you had started analyzing your images concurrently as they're being captured. The system is only minor slowdown in the analysis section.
Great. Just give people a minute, see if we get some additional questions. All right. I'm not seeing any additional questions come through. If you are watching this as a recording, or if you do watch this again after the fact, or if a question comes to you after we've concluded today, certainly feel free to reach out to us at support@nexalom.com. We have a great team of technical support specialists, customer service representatives, and Celigo experts. So by reaching out to support@nexalom.com, the appropriate department can pass you along to the appropriate Celigo expert to assist you with your questions. So unless anybody has any final questions or comments, I think we can go ahead and wrap this up for today. I want to thank